Good afternoon, everyone. Welcome to the final session of this workshop. I hope everyone can hear me. I would like to introduce our first speaker for this afternoon session. Please welcome Ghanim Ullah. I met Ghanim Ullah 8 years ago at a biophysics conference in San Diego. And I suppose we were fierce competitors at that time because we both published models on the IP3 receptor at exactly the same point in time. But there we were in this gigantic hall full of hundreds of posters, literally, and I have to say that I had the best time at this conference with Ghanim when we explained our models of the IP3 receptor to each other. At that time, Ghanim was a postdoc in Los Alamos with John Pearson, but I should really start with the beginning, I think. Ghanim received his bachelor and master's degrees in physics from the University of Peshawar in Pakistan. He then moved to the University of Ohio and did a PhD with Peter Young in 2006. He was then a postdoc with Stephen Schiff at Pennsylvania State University before he moved to Los Alamos to John Pearson's group. And since 2013, he is at the Department of Physics at the University of South Florida, where he currently is associate professor. I will disappear now and let the stage open for you. I'm looking forward to your talk.
Thank you. Okay, thank you, Ivo, and congratulations for putting such a wonderful workshop together. It was long overdue. So I'm going to talk about something which is slightly different than what we have been hearing in this workshop so far. It's amyloid beta pores, and if you don't know what they are these are peptides that are accumulated in the brains of Alzheimer disease patients. And they do all kind of bad things to the brain and specifically to the calcium signaling in different ways. So I'm going to start with the contributions. All the experiments were done by Angelo, who is at University of California, Irvine. Dr. Shah did the software development and the modeling. And he's a postdoc in my lab, and the work is funded by NIH through this grant. So what are the questions that we hope to address with this work? As I said, amyloid beta are the main hallmarks of Alzheimer's disease. And they target several aspects of calcium signaling. What we want to find out is that what are the key pathways that are involved in the calcium impairment. So calcium signaling is impaired in neurons and astrocytes and microglia, everything that you name it, it's impaired in Alzheimer's disease, and we want to know what are the pathways for that. And then what are the primary downstream targets for these impairments? So those are basically the two main questions that we are trying to address. So what do these amyloid beta do? If you take some oligomers of amyloid beta and you apply them to the extracellular space of the cell, neuron or any other type of cell, and you wait for a few minutes, they will make pores in the plasma membrane. Actually, they can make pores in any membrane, plasma membrane, the ER, the mitochondria, all the intracellular organelles. And those pores are permeable to calcium and other cations. But we are interested in calcium, so I'll focus on that. If you apply them intracellularly, if you inject them into the cell, they can generate IP3 through the PLC pathway. And the extracellular A beta can do the same. They go inside, we believe they go inside, and they generate IP3. Similarly, the intracellular A beta can make pores in the plasma membrane and the other membranes. So now, if you look at through a microscope, if you look at a piece of membrane, this is what you see. These pores open and close like normal ion channels. They have very interesting properties. and they evolve over time, and they bring in a lot of calcium to the cell. So for this talk, I will mostly focus on the amyloid beta pores. We have done some work on the PLC pathway as well, but that may be some other time. So before I tell you about the data and the modeling of it, I want to tell you the experimental protocol, kind of a cartoon version of it. And I think this is really important to this. This slide is probably the most important slide in the talk to understand why the modeling is a little challenging. So when we apply amyloid beta, we wait for about 20 minutes. The pores are formed. And then we image a membrane, typically 40 by 40 micrometer size membrane. And we make a movie of that membrane for about 30 seconds, where we have about 15,000 frames in the movie. Now we have to, ideally, we want to image these pores for at least an hour or longer to see their long-term evolution, because they do evolve over time. But we cannot do that because that would damage the laser would damage the membrane. So we wait for 5 minutes, we make another 30-second movie, we wait for 5 minutes, make another 30-second movie, and we keep doing that for as long as we can do it, and as long as the cell survives. So we have a gap of information here. We basically look at the pores for 30 seconds, and then there is this gap that we have to wait and then observe the pores again. And things happen during those five minutes. A lot of things change, as you will see. So what we are interested in is what happens to the number of pores over time. For example, if we see 100 pores here we might see maybe 200 or 50. The number doesn't remain the same, and so that's the one question that we are trying to understand or address. What are the gating properties of individual pores at a given time? If we analyze the pores at this point, for example, how they behave, how they gate, and then how they evolve when we move from one movie to another, how their gating properties change over time. So those are the questions that we are trying to address. So this is basically three-step process. Step one, amyloid beta pores are extremely small. The flux through them is typically smaller than your usual ion channels, and they have very low open probability. So that makes it even harder to detect them, because you see them about in every 100 milliseconds, you will see them for 5-6 milliseconds. They will be open for 5-6 milliseconds, and then the rest of the time they are closed. And there are a lot of them. If you wait longer, you will see thousands of these pores in a 40 by 40 micrometer membrane. So we needed automatic methods to detect, to locate basically where these pores are, and then analyze them in a more efficient way. So to do that, we developed this software called CellSpecs. The details are in this biophysical journal paper that came out in 2018. But I'll just briefly tell you what this does. So when you load your movies, are TIFF images, the movie state, it does a few steps to identify the channel locations. And those steps are basically explained in the paper, so I'm not going to talk about that. It will identify all the channels in the membrane, and then you can investigate single channels. You can just click on individual channel in this window, and it will show you a time trace, the idealized trace for the gating of the pore. It can also it makes a location map of all the pores, and at the same time you can also save all the time traces for all the pores in the membrane. So it doesn't matter how many there are. There could be thousands of them. You just click a button and it will save all the time traces for all the pores in the movie. and then it generates some nice statistics that you can use for, just looking at what the pores are doing. You can use them later for modeling and so on. Like the open duration, for example, the mean open duration of the channel. And as you can see, they are typically open for a few milliseconds, 10 to 15 milliseconds, most of them. Their closed duration is extremely long. It can go up to 4 seconds. And the open probability is, as I said, it's really low. And then the maximum amplitude is, they have a wide range of properties, basically. But it's really easy to extract them using cell specs. So one interesting, if you are when you are a modeler, you kind of look for these interesting things, is that these channels, they have multiple conductance levels. So you have an open state, it will open to first conductance level, second, third, and so on. And it can go up to 6 conductance levels. I have shown 5 here, and I'll show you in a few slides how we get these idealized traces, where we take the time trace, which shows the fluorescence signal, to basically extract just the idealized trace. But they have multiple conductance levels. Their open probability varies a lot. And as time goes by, you will see that they will go from a state where initially they would open in 1 conductance level to maybe 2, 3, or even 5, 6 conductance levels. So they are really interesting to study in modern. But as I said, they evolve over time. So, for example, here in this case, we tracked 5 pores over time. So in the first movie, as I said, after 20 minutes after adding the amyloid beta, we made a movie of 20 seconds in this case. And you can see that these 5 pores, 2 are completely silent almost, and then the other 3 are showing some activity. But as you move in time, 30 minutes, 35, by the time you reach 60 minutes, they show a lot of activity. Their open probability is big. Their mean open time is longer. Mean close time is shorter. And this is a general trend in them. They basically evolve. They get bigger, more toxic over time. So another thing that we can do with cell specs, as I said, you can generate a location map where these channels are in a given movie. So we generate a location map for one movie, and then we wait 5 minutes. Then there is the second movie. We generate a location map for that. We keep doing that, and we can put these maps on top of each other to identify the pores that appear in multiple movie stacks. Some of the channel would just appear in one movie, and then they would completely disappear because we know their location. In the experiment, we don't move the apparatus, we don't move the cells. Everything is fixed, so we know that a pore appearing in the first movie, where it should appear in the second movie, and so on. But there are pores that will appear in 7, 8 movies, and there are some which will just appear in 1 and disappear. So we can study their properties individually. The pores that we call common pores, common between different movies, and the pores that are unique, which just appears once and then disappear. So if you look at the mean open time, for example, so I have 2 different types of bars here. One is red, which is all the pores, including the unique and common ones, and then only the unique pores. They both show an increasing trend in the mean open time. As you can see, so on the x-axis, I have the time of recording in minutes. They start with relatively low, smaller mean open time, and it increases. Same thing happens for the mean open probability. Both pore types, the unique and common, their mean open probability increases. Their maximum amplitude, the maximum flux that you will ever observe through that pore, increases over time. So That you and then you can also look at pores that, for example, are common in two movie sticks. When you see them the first time, they will have relatively smaller amplitude. You see them later, the amplitude gets bigger. Same thing goes for mean open probability, the mean open time, all those things that you are interested in. And if you have them in three movie stakes, you see it's the increase in the amplitude and the open probability is persistent. So and there are some questions that we are still trying to understand, is that these unique pores, where do they go? We believe that though they go inside the cell, but that's something that we have to be certain and figure out yet. But anyway, they do have really interesting properties. So and they the bad thing about them is that they are more active at the resting membrane potential. So if you look at if you record the fluorescence or the flux to these pores at 0 millivolt membrane potential, there is not much going on. Minus 40, a lot of flux going through, and minus 80, which is close to the resting membrane of the neuron. Huge flux, calcium flux through the pores. And so here is your plot, the fluorescence amplitude or the flux, the average flux through these pores. Around minus 80 is when it's maximum, basically. So that's a bad thing, because that's where the neuronal and the astrocytic membrane potential is sitting, and you really don't want that to happen. Zinc turns out to block these pores reversibly. So this is A beta 42 at minus 80 millivolt. Just 6-7 examples from the pores. You apply zinc at under the same conditions. A lot of these pores are silent. You just see some noise, but most of them are silent. And then you can look at all the pores in the experiment in the membrane. So in this case, we have along this axis, we have the pore number starting from 1 to more than 400 pores in this experiment. And then on the x-axis, we have time. And each bright spot represents, it tells you that the pore was open at that time. So as you can see, there's a lot of activity going on over time in the membrane. When we apply zinc, most of the activity disappears. You wash out zinc, the activity appears again. So they are blocked by zinc, and it's a reversible effect. We believe that. Okay, so what is the second step now? We are trying to move towards the modeling of it. So when we talk about modeling single channel data, we basically need idealized traces which tells us open, close, open in one conductance level, 2, and so on. And then we need dwell times distribution. So it's if you want to do this for single traces for thousands of pores, it's a daunting job. So we developed another automatic software that we call TraceSpecs. So it's a series TraceSpecs after CellSpecs. But this is again, it's a really nice software. You just click a few buttons, you load your trace, a noisy, it can be fluorescence trace, it can be from page clamp experiments. It removes the drift, the background noise, and gives you nice idealized trace. And you can look at the trace by investigating and just observing it. And if it doesn't make sense, you go back and redo it, just change one parameter or the other, and it will fix it for you. And if you like running codes in the terminal, you basically can run it over, we have provided the software and the source code with it in the publication. So you can run the code on thousands of traces in one go. And what you get at the end is these idealized traces, things like the mean open probability, mean close time, open time, and the dwell time distribution, which is what you need for modeling. So it's another useful thing to have. So as I said, you take fluorescence traces, you get idealized traces. These spores, they evolve over time. They will initially have low open probability, low amplitude, and then the amplitude and open probability increases over time. So we take these idealized traces. Oh, and then, as I said, you generate the dwell times. So, for example, the dwell times in the conductance level 1, for example, would look like this. Conductance level 2, 3, 4, and so on. It doesn't matter how many conductance levels you have. You could have more than 5 or 6, and it generates these distributions for you. So then we have the third step, which is we take these traces and the distributions and model them. So that's what we do. Just to quickly remind you that there is an information gap in the experiment. So we will model the pores activity at this time, at 20 minutes after the application of A beta, and then we will model it again at 30 minutes, 25 minutes, and 30 minutes, and so on. And then we need to merge those models to get an idea that how they evolve over time. So each moving frame is modeled by a separate Markov chain model, and then we put them together. Okay, so you can do this since these models are the rates are not really complicated. They are basically constant. So you can use QUB for modeling them. You can write your own subroutine, use things like maximum likelihood to estimate these rates. And the way we do that, we start with the simplest possible model. So we know there is one closed state, and we know there are up to 5 open conductance levels. So we have one closed state, 5 open states. And then we fit this model to the dwell time distribution in closed state and in the dwell times in all open states. And we optimize these rates. We also try to make the model a little complex to see if the outcome improves. And if it improves significantly, we keep the model. Otherwise, we just stick to the simple possible model. And in most cases, basically, the more complexity doesn't add much to the model. So we just stick with the simpler model. So, and the lines here, so the distributions are basically the red is the experimental data, and the lines are the fits from the two models. And then you can also look at compare the open probability for each subconductance level. This should be subconductance level. Mean open time, and see how the model compares. And it compares really well because it's basically the model is derived from the data, so it shouldn't have huge discrepancy with the data. Okay, so we get these rates, these forward and backward rates at different times after we start the experiment. Then, so we will have, if we have 10 movies, for example, we will have 10 values for each rate constant at different times. Then we take those and we fit some simple functions to these rate constants. So now you have the rate constants are a function of time now. So if you want to model the pore 20 minutes later, you will have one constant. For example, this is the forward constant between closed state and subconductance level 1. You will use this value. Otherwise, you basically take the value corresponding to the time from the point where you added your amyloid beta. So, and this is clearly, it tells you that they do change, that their kinetics change over time. So we get these rates, and now we can model them basically at any time. It doesn't matter if you want to look at their properties 10 minutes later or 10 hours later. You have the model which is based on the data. And then you can do all kind of cool stuff with it, which is what we are beginning to do now, is to put them in the membranes and, synapses, and see how they affect the function of these different cell functions. So the model, it closely follows, which is no surprise because everything is based on the experiment. It follows the experimental data. The mean open probability is a function of time, the mean open time, the mean close time, and also the probability, the mean open probability in each conductance level. So we have 5 subconductance levels, and we basically it does a really good job in tracking the dynamics or the kinetics of the pores. One thing that I want to point out here is that this is the subconductance level 1, and I believe this is the open probability of the subconductance level 1, and it increases over time. Actually, the open probability of each conductance level increases over time, but if you compare the individual probabilities, here it's 0.01 and goes to 0.02. The higher conductance level are very rarely visited. So the toxicity increases over time, but we believe that it's mostly because the open probability, or the probability to visit the lowest subconductance level more frequently, is causing this toxicity. So that's basically all I have. I'll just summarize. The amyloid beta oligomers form pores in the membrane with a wide range of gating properties. I hope I have convinced you of that. These pores have multiple conductance levels. The maximum conductance and open probability of pores increase with time. Pores become more toxic over time because their conductance, probability, they all basically increase over time. And zinc can block these pores reversibly. But for us, I think the most important thing as a modeler is that this gives us an opportunity to model ion channels in their native environment, because the imaging experiments are done on these pores or channels in their native environment. So I think we think this is a really powerful approach to see how the channels behave in their native environment. So I'm going to stop here and take questions.
Thank you very much, Ghanim, for your really nice talk. I have 2 questions from the audience. So the first one from Maurizio. Ghanim, I might have missed it. Could you say a bit more clearly how the software helps detect the pores based on the calcium-dependent readout, I assume? But this only works in cultures, I assume. How can I debunk pore activity in real tissue, where likely extracellular calcium homeostasis is an important player in the regulation of such pores? So the question is a bit lengthy.
Yeah, no, I mean, it's a valid question. So it's a good question. And so the first thing that you have to do as an experimentalist, you have to make sure that what you are imaging is basically the channel that you are interested in. And that's what we made sure. We basically pharmacologically blocked all the channels, and then another test was, since these were the channels in the plasma membrane, we removed the extracellular calcium and made sure that the fluxes disappear if there is no extracellular calcium. And the driving force, zinc, is another test where we apply zinc and the channels disappear. So the software, basically, to answer your question, it wouldn't be able to tell you if you see 2 spots. It wouldn't be able to tell you if that spot appears because of voltage-gated calcium channel, for example, or amyloid beta pore. Because all it does is it looks for the intensity, the fluorescence. So you have to, in the experiment, you have to first separate the channels that you are imaging, and then the software basically it just looks at the intensity. So to kind of differentiate multiple channels, and that too, you will have to know their properties very well. You could probably use something like machine learning tools, but not this software. It wouldn't be able to differentiate between the two.
So, Ghanim, there are further questions, but the problem is that we have run out of time a bit. So,
Okay.
I would like to invite the next speaker on stage, but I thank you for your talk. And if you like, you can answer the questions in the Ask a Question field and possibly also on Neurostars. Thanks a lot. I have now invited on stage our next speaker, Silvina Ponce Dawson. Silvina received her PhD in 1988 from the University of Buenos Aires in Argentina in theoretical plasma physics, and in her research career she has made contributions to statistical mechanics and nonlinear dynamics. She has a long-standing interest in spatiotemporal models of calcium dynamics, which will also be the topic of her talk, I think. For many years, Silvina has served the physics community in the International Union of Pure and Applied Physics, UPAP. She led the Women in Physics Working Group, for example, is the vice president elected at large with gender champion duties of UPAP, and most recently she has been appointed to the acting president designate of the UPAP. Currently, Silvina is a professor in the Department of Physics at the University of Buenos Aires in Argentina. I'm looking forward to her talk.
Okay, thank you. So let me share an application. Okay. Thank you. Can you hear me? Is it okay?
Yes, we can hear you.
Okay,
Very
good.
nice, thank you.
Okay, so I prepared a talk that was research was originated in behavior in neurons, particularly in synaptic plasticity, although I've been thinking of other things afterwards. And this is work that we've done mostly with Ana Calabrese. I am also going to show some experiments that Cecilia did, and then Herman is now taking over the work of Anna's. Okay, so this is about synaptic plasticity that we've heard quite a bit yesterday, and in which calcium plays a very important role. And the efficacy of the synaptic transmission depends on the activity and can be modified. And the examples, experimental examples, are what are called long-term potentiation and depression. And this means an increase or decrease in synaptic efficacy that can be elicited in different ways. And one way is stimulating the pre- and the postsynaptic neurons almost simultaneously, but with a certain delay, and depending on where you apply the stimulus first, you then have potentiation or depression. So we've heard about this yesterday, and this is the plot where you see, depending on the relative timing of the pre- and postsynaptic stimulation, which is the training that is going to change the synaptic efficacy, then when you look at how the synapse responds to one stimulus, you see that it can be depressed with respect to the level before the training, or it can be potentiated. And well, this change in the synaptic strength can last for weeks. Here is shown only for 50 minutes or something. Here you have LTP. This is how the response looks like in some units before the training. Then you apply the training where the arrow is here. And then you again apply the same stimulus as before and see what the response looks like. And you see it either enhanced or depressed. And sometimes you don't see any change. Anyway, and there are many factors that participate in this change of the synaptic strength. And one important factor is calcium release from the endoplasmic reticulum, which is depicted here, through IP3 receptors and also ryanodine receptors. And both ryanodine receptors and IP3 receptors are subject to what is called calcium-induced calcium release, that is depicted here for the case of IP3 receptors that need not only calcium bound to the receptor to become open, but also IP3, so two things bound to the channel for the channel to become open. And so here they are on the membrane of the endoplasmic reticulum. And if you have IP3, and so IP3 is bound to the correct sites of the channel of the receptor, then when calcium is also bound, the channel becomes open, calcium is released, and the very same calcium that is released diffuses and can act upon other channels. And so you can, other channels that have IP3 bound, and then you can have this propagating calcium wave. Now, in the case of IP3 receptors that we've seen some models today, cytosolic calcium plays a dual role because it's activating at low concentrations and inhibitory at high ones. And the simplest kinetic model, sorry for this drawing, which is very bad, but it's to illustrate the thing, would be something like having one binding site for IP3, and then two binding sites for calcium, one of high affinity, and so that calcium binds with high probability even at low concentrations, and the other of much lower affinity, so that you need a much higher calcium concentration to bind to that site. And if calcium binds to the high affinity site and IP3 is also bound, then the channel can become open and calcium goes through. While if calcium is bound to the low affinity site, then the channel is inhibited. Anyway, another key component of synaptic plasticity is other types of receptors that are on the plasma membrane of the postsynaptic neuron, particularly NMDA receptors that are also calcium channels, but they are usually blocked. Their pore is blocked by magnesium, and so you need to change the membrane potential of the postsynaptic neuron for the magnesium to be kicked out of the pore, and so calcium can get through. So that's why you have some coincidence thing. It's like a coincidence detector, the NMDR. And then LTP and LTD is associated to changes in the level of cytosolic calcium in such a way that, well, this would be like basal calcium, so you don't have any synaptic modification. But then if you have some intermediate calcium concentrations, you have depression because you activate some phosphatases that turn down the conductivity of some channels, or even they can let them get invaginated. And then if you have higher concentration, then you potentiate those channels. It's kinases that act on the channels, and so you have the potentiation of the synaptic strength. The thing is that because calcium does not stay in a region but can spread about, not always, because there's these buffers that don't let them go very far away from its point of entry, but you can regenerate the signal through calcium-induced calcium release, then the synaptic modification does not stay at the homosynapse. It also can change the heterosynapse. And so other synapses, not the ones that you trained with these protocols that lead to either LTD or LTP. And in particular, this is important because if you have excitation so synaptic strength that is becoming more higher and higher, then that can be, you need something to compensate for that, for the whole network to be stable. And apparently these heterosynaptic changes can help normalize and stabilize this strength. And in these heterosynaptic changes, calcium release through IP3 receptors plays a role. And so the work we looked at with Anna is a work which is an old work, but it did all sorts of experiments that are very interesting. So this is the schematic scheme of the type of experiments they did. It was in hippocampus. And so this is the postsynaptic neuron. And this is what is called the homosynaptic pathway, and this is the heterosynaptic pathway. And in the upper box, I'm going to illustrate what happens in LTP, and here what happens in LTD. And basically, if you put some stimulus in the homosynaptic pathway, which is this red thing here, then you see this type of response in the postsynaptic neuron. If you do the same in the heterosynaptic pathway, you see this response. This is before you train you do either the ELTP or the LTD protocol. And so the LTP and LTD protocol in this case means that you apply some stimuli, I don't see my here, on the postsynaptic neuron and the homosynaptic pathway with a certain order. It could be that the postsynaptic neuron comes after, or it comes before. And so in one case, you will have LTP, in the other, you are going to have LTD, and this training means tetanic stimuli, so it means with a frequency. And so then you again apply the same stimulus that you had applied before the training, and you see what's the to the homosynapse and to the heterosynapse, and see what's the response. And in the case of the LTP protocol in this paper, what they observed, of course, they observed LTP at the homosynapse, and they didn't observe any change at the heterosynapse. And in the case of the LTD protocol, they observed LTD at the homosynapse and LTD at the heterosynapse. So these are some of the results that were obtained in this paper. They also blocked type E3 receptors and ryanodine receptors. And from those experiments, they arrived at various conclusions. I'm going to quote only two. One is that the P3 receptors, the activity of the receptors determines this, what they call the polarity of the synaptic change. By this, they mean that LTP is restricted to the homosynapse, while LTD propagates from the homosynapse to the heterosynapse. And then the release from ryanodine receptors is also very important for the induction of LTD. And so they produced in that paper this, what we call a qualitative model, in which they say, well, this is a spine, and this is the dendrite, and this is another spine. So this is like the spine that you trained, and this is the heterosynapse. And you see, this is the membrane, the plasma, the membrane of the neuron, and this is the endoplasmic reticulum in pink, sort of. And so you have all these players here. In particular, you have the ryanodine receptors localized in the spine, but then the IP3 receptors are all along the dendrite, so that the IP3 receptors allow for the propagation of a calcium wave, and so that calcium can spread by a regenerative process from the homosynapse to the heterosynapse and change the synaptic efficacy afterwards. Okay, and this was confirmed by other works afterwards. So we wanted to understand this propagation, and to this end we developed two models. One, a model for the dynamics of calcium in the homosynaptic spine, and then a model for the propagation of the IP3 receptor mediated calcium wave. And so putting them together, it's a one-dimensional model in space in which we are going to have, I'm sorry, some of the figures are in Spanish though. Anyway, so this is the spine, and where you have calcium entry from the extracellular medium, and then this is going to diffuse. And also at the spine, you will have production of IP3 through metabotropic receptors that when the neurotransmitter gets bound to them, then they start to produce IP3 from precursors on the plasma membrane. And so then you will have the a propagation of the wave or not. And actually, this is sort of an image of the dendrite and the spines that can be different sizes stuff, but we are going to have this one-dimensional model. So for the spine model, we pretty much took what Chauvel did in 2002, but we added something to account for some of the observations of Nishiyama et al., which was this release through ryanodine receptors, and also potassium channels that are calcium modulated, which are these SK channels. So basically, Chauvel's model, for them, the calcium dynamics is mostly due to the current through the NMDA receptors, and then there is some removal. And then for that, in that current, you have something that is related to the synaptic conditioning and the postsynaptic conditioning that is going to change what the conductivity of the MDA receptors is, basically. And then there is something that is related to the membrane potential of the postsynaptic neuron. And this is what we changed slightly with respect to Chuval's. Chuval's considered the backpropagating action potential, something through these other receptors that change the voltage to kick out magnesium ions from MDA pores. And then we added also some change due to these potassium channels that are modulated by calcium. And for that, we used some experimental results to model that part of the contribution to the postsynaptic potential. So with that, we obtained the different behaviors in the dynamics of calcium depending on if it was LTP or LTD. And then we put that into the, we wanted to couple that to the propagation. And for the propagation, we used a diffuse fire model, basically, where U is the concentration of calcium with an effective diffusion coefficient, number of ions that are released per unit time, and that's a stochastic. And then the channels, the receptors are located in particular points that are separated by 2 microns, by D is 2 microns, and then you have calcium pumps. And for the release of calcium, we produced a stochastic cluster model that we constructed based on observations of Ian Parker's group. Basically, it was a model for the cluster, a closed state for the cluster with a transition to the open state that was proportional to calcium and modulated by IP3, but basically a threshold. And then the release was also considered to be stochastic because we are assuming the cluster is a bunch of channels, so we don't know what the current is going to be. So that's taken from a distribution. And then 2 inhibited states, one that is calcium modulated and the other one that is not. And we put that into our one-dimensional model of the dendrite with 15 channels. And the initial condition was everything was basal. The clusters were closed, 15 clusters actually. And then we put the spine model at one end, and we considered that the heterosynapse was like 500 nanometers away from the last cluster, which was the 15th one. And the domain of the integration is double the domain where the clusters are. So first we studied with uniform IP3, and we couldn't reproduce this observation that LTP was restricted to the homosynapse and that LTD propagated to the heterosynapse. So the way that we look at is by looking at calcium at the homosynapse and at the heterosynapse after when we apply one or the other protocol. This is for the LTD protocol, and this is for the LTP protocol. So we have an enhancement of the calcium concentration in the homosynapse, but at the heterosynapse, we also see an intermediate increase of calcium. It would be LTD and not basal. And so then we thought, well, IP3 is being produced at the spine, and we were thinking that IP3 was uniform everywhere. And so we added so we said, how are we going to change that the LTP, which means high calcium concentration, is restricted to the homosynapse, and it's low and basal at the heterosynapse? When you increase to intermediate value at the homosynapse, you also have intermediate at the heterosynapse. And so the way that we thought of was that we had to include the dynamics of IP3 because calcium plays this dual role. So we thought if calcium arrives at the clusters before IP3 does, then the clusters might go into an inhibited state. And then the wave is not going to propagate. And that's more likely to happen in the case of the LTP because of the higher calcium concentration. While if you have moderate levels of calcium, then it's not so probable that calcium will bind to the inhibitory sites of the channels, and so you will have the propagating wave. So the relative time at which IP3 and calcium arrives at the clusters determines whether the waves can propagate or not. And the way we included that was a very simple way with a diffusion equation for IP3 and with a coefficient that we took from Albritton, which is 250 microns square per second. And the way that we could reproduce, at least more or less qualitatively, the observations was assuming that IP3 was delayed in its production by 500 milliseconds. And so then we could reproduce that with the LTD protocol. We had intermediate increment at the homosynapse and also at the heterosynapse, and then large increment at the homosynapse for LTP and almost basal at the heterosynapse. And these are at the heterosynapse is a mean over 100 realizations. So this, whether the wave propagates or not, depends on whether IP3 or calcium arrives first at the clusters. And so I'm going to connect this with other results. And in particular, in this paper, they say that these IP3 receptors are coincidence detectors because they need to detect the mobilization of IP3 and the postsynaptic calcium signaling at the same time. And what we see in our studies is not just the coincidence, but also the relative timing of building up one concentration or the other and how it gets transported into the cell. And so this is a very delicate balance that can be changed in various ways by buffering, and also if ryanodine receptors participate of the release in certain points along the wave. And well, this form of training the synapse with this delay of the timing between what you do to the pre and the postsynaptic neuron, it's been observed that you produce different things depending on the cell. So this is because of this very delicate balance that you can have different behaviors, basically. And this is because of the dual role of calcium waving on IP3 receptors. And it's interesting that some other works have seen different behaviors depending on whether you elicited some response with IP3 only and with basal calcium, or if you pair that response with calcium entry due to action potentials. And finally, I want to mention that the values we used of the diffusion coefficients come from this very old paper by Albritton et al., which is from 1992, I forgot the year, in which they say that IP3 diffuses at 280 microns square per second, that calcium is strongly buffered. So here you have an effective diffusion coefficient as a function of the free calcium concentration. And the maximum value that they say that calcium could reach in its diffusion, which is the free diffusion coefficient, is 220 microns per second. But in recent works with Ian Parker, we observed that IP3 seems to be also buffered. These are experiments in which IP3 is caged and released with a UV flash that is either localized or is like spread along the cell. And what you see is what puffs are evoked by one or the other kind of release in IP3. And in particular, what we looked at was the latency to the first puff. And in the case of a localized release of IP3, you see that the latency to the first puff depends on the distance, while if you have the spatial spread of the release of IP3, the latency does not depend. Now, if IP3 diffused at 280 microns square per second, then there shouldn't be such a large difference between releasing the IP3 at one point or having it spread all over the cell. And so we tried to fit what was the best diffusion coefficient that could account for these differences, and we found between 5 and 10 microns square per second, so much smaller than the value of Albritton's. And then regarding calcium, with Cecilia, we've been doing experiments, FCS, fluorescence correlation spectroscopy experiments in Xenopus levi zoocytes and in yeast to determine the diffusion coefficient. And basically, in FCS, you take the autocorrelation function and try to fit it with different things. And when we fit with two components, there is one that has the largest diffusion coefficient. That was for in the oocytes. It had a peak. It had like two components itself, but so it looked bimodal. This is histograms of the values we obtained from the fits, and they are around 300, 400 micron square per second. And in yeast, a little bit smaller. This is yeast under basal condition. This is yeast in high calcium, and then you see that other bump there in the distribution that we have to interpret.
Silvina, you have around 5 minutes. Yeah, okay. You were
I am in the summary.
Thank you.
So basically what I tried to show you was Nishiyama a long time ago showed that calcium release from internal stores through, the stores went away. Anyway, through IP3 receptors plays an important role in determining the specificity. So that LTP stays in one homosynapse and does not propagate, while LTD does propagate, and the polarity of synaptic modifications. The simulations of our model, that is a huge simplification, indicate that maybe this is related, the specificity of LTP and the non-specificity of LTD, to the dual role of calcium on IP3 receptors and whether an IP3-mediated calcium wave can propagate or not. Then, whether it can propagate or not depends on the relative delay with which IP3 and calcium reach via channels that have to become open to regenerate the wave. And that depends on how fast they diffuse. And based on the recent experimental results, we are going to reanalyze these simulations with new diffusion coefficients. And before I stop talking, I want to thank the organizers, and I wish we could have met in Melbourne, that I was going to combine this workshop with attending the IUPAP International Conference on Women in Physics, which was the 7th in a series. This is a picture from the previous one that is attended mostly by women, but also men. And there we try to make people aware of what women do in science and try to make visible their work. So this is not to be taken as a recrimination, but just a recommendation to think, because I would have liked to see more women giving talks in this workshop. And this is something we've been also discussing quite a bit, not only in physics, but in all STEM areas. And this is a book that we produced with a project, an international and interdisciplinary project. The book is downloadable and has results from a survey that was answered by 34,000 people from over 130 countries, people working in STEM. And so this book is just a call of attention to say that we do science. And given that we were going to have this workshop in the Southern Hemisphere, which is not that common, let me tell you, and I come from the Southern Hemisphere, it's good to turn things a little bit around, to look them from a different perspective, to make not only the world, but also the practice of science a more equitable endeavor. Thank you.
Thank you very much, Silvina, for your very nice talk. Are there any questions? I think I see one popping up. So from Martin Falke, he asks, nice talk. Do you get the dendrite heterohomosynapse also with a small IP3 diffusion coefficient?
Well, we haven't tried that, but it's what we are starting to analyze. And I am very excited basically because the way in which we could reproduce that different behavior before was by delaying the production of IP3. And so I hope that maybe with the smaller diffusion coefficient, you don't need to introduce this delay in the production of IP3. So that's what I would like to study in more detail.
Thank you very much. Are there any further questions? Well, I mean, it's still possible to ask questions to Silvina in the Ask a Question box. I would now like to invite our next speaker and also Maurizio, who will introduce him. But I would like to thank Silvina again for her nice talk. Thank you very much.
No, thank you. Okay, bye-bye.
Can you hear me?
Yes, I can hear you.
Very good. So Ivo, you are okay? Very good. You can invite now Evan. I have the pleasure to introduce this brilliant young investigator, Evan Creswell-Clay, who I had the pleasure to meet last year when I was actually popping in Florida State University for a visit to his supervisor then, Gordon Lebacher, who I don't know if he's in the audience right now, and if he is, I say hi, or we say both hi. So I came across Evan's work during his latest years of graduate studies, as he was starting an approach, a new one, on how to model calcium signals in astrocytic processes, so the usual compartmental modeling. So he started that model, and because he was kind of inspired by some ideas that I had myself in that direction, turns out that I had the pleasure to be a reviewer of his work, of his final thesis work, not only on the thesis but his first article. And then with that in mind, I said, why don't we visit this guy and his supervisor? And so I pop up in Florida, where humidity was, however, good enough to keep me there and have good time during the last summer. And so we had a nice chat, and we said, why don't we start collaborating and think about something else and some evolution about this work. So I'm very glad, because Evan eventually, was about to finish. So, it's finishing the PhD. He was very stressed. And he had to finish up the results, and then he said, "Oh, thank you. Arrived. You gave me basically the results for my final thesis." It was a little bit idolatrizing the one who is speaking, but we said, Evan, why don't we just manage to think about working together in the future? And so we wrote together a Fulbright application, and turns out that we won this Fulbright application. And I don't know if in the audience there is also the third collaborator that Evan who will actually introduce, James Schumers, who is at Florida International University, and some of you may know. So anyways, without any further hesitation, I will let Evan speak. Okay, so it will show mostly some ideas and some concepts or early preliminary work that we are about to develop. And also, I would say that right now, Evan is at NIH, at the lab, at the division led by Arthur Sherman, and is actually working on some machine learning and protein folding modeling. So he's really developing a multidisciplinary expertise. And with that foot on multi-protein folding, whatever, and now he's dipping back on calcium and showing you some ideas in that respect, and in the direction of calcium compartmentalization. I was way too long by now, so I will let Evan share his presentation and start with his talk. Good luck, Evan.
Thank you, Maurizio. And I want to start by saying that I am really honored to be here. I'm really excited to talk to you all about some preliminary work for that Fulbright grant that I was awarded. So, yes, good afternoon. Most of you are it's the afternoon for you. So I'm Evan Creswell-Clay, and I'm going to be talking to you about some compartmental modeling that we have been doing, incorporating astrocyte physiology and morphology. So I want to start by acknowledging as well the Fulbright and Maurizio for his invaluable assistance toward the end of my doctoral work, as well as for the Fulbright application that we put together. I also want to thank, again, Gordon Erlbacher, Florida State University, my PhD advisor, whose influence you're going to see in some of the work today as well. And finally, I want to thank James Schumers at Florida International University, FIU, whose experimental work is going to be pivotal moving forward as we carry out this Fulbright Fellowship starting in January of next year. So I want to talk to you all about calcium dynamics and astrocytes. And before we go into that, I want to show you this image from the lab of Emmanuel Beaurepaire. And we're seeing here from left to right different scales of a cutout of the cortex. And this fluorescent imaging is showing us, as we move into these smaller scales, this almost galaxy of multicolored cells. And in these cells, on the right-hand column, you can see these stars, these bright spots, the neurons, which are engulfed and surrounded by these tessellating cells, astrocytes, which can be characterized by their very interesting and complex morphology. And one of the big questions in the last 20, 25 years has been, how do these cells function and how do they operate and what is their influence? And one of the things that I want to be considering is how does the morphology of these cells influence their activity and their function? And when we're talking about astrocyte function, we really need to discuss calcium dynamics, because calcium is the functional readout, the excitable readout out of these cells. So as an example, I want to show you some work from James's lab where he is taking a mouse and he is giving it some type of visual simulation, in this case, an oriented grading. And he's recording from an astrocyte in the primary visual cortex as he's changing this stimuli. And as we go down through these different calcium traces, which are being recorded from different subcellular region of the same astrocyte, we can automatically notice one thing, which is on the right-hand side of the traces, this universal response to this specific green orientation, this green grading. A response that is not necessarily seen in the first part, in the other orientation of that green grading, where you only really see an elicited calcium response in this bottom row for this peripheral process. So this is kind of a salient example of the type of subcellular calcium dynamics that you can see within these cells, which in this case are responding in an orientation specific manner to different types of external stimuli. And a work that really postulated this very well, the first work to really postulate the seminal work by Volterra and et al. In 2014, was to look again at a cell, at an astrocyte with these two processes here and we can see in the top row two different types of characterization of the calcium activity going on in the cell. One on the left hand, the top left, is the cumulative calcium activity within the cell, as well as the frequency of calcium events that we are seeing. And this mapping between these two is not one-to-one. We are not seeing a complete correlation between the two events. We're seeing a lot of activity in the soma, cumulative activity, whereas the frequency is not as strong as compared to some of the other areas in the cell that, while cumulatively active, are showing much higher frequency. And when you combine these two perspectives, you can start to create this spatiotemporal map of the cell and divide it into these different calcium microdomains or subregions, which could potentially express different types of dynamics. For instance, going to this bottom right-hand image, we can have the soma with these high amplitude, maybe less frequent calcium events, while in the periphery in these processes, we're seeing more frequent calcium events with a little bit less activity. And this work is really showing us that we can start to characterize the activity in these cells through these subcellular domains, through these microdomains. And we want to try to understand their interaction as well as characterize the events within them. But as far as characterizing these different events, we have to incorporate multiple features. And I want to bring your attention again to a astrocyte with a soma and two processes in this great work by Bindacci et al. In 2017. Where we're looking at, again, calcium activity within these two processes in the soma, as well as, we have some activity in the end foot and some activity in the glial pill of the cell. But what we can see is that, yes, you have this subcellularly confined events, but it is very heterogeneous, and it's not at all well defined based off of the location within the cell having some type of consistent activity. And this can be seen in this right-hand figure in the top row, where when we model the activity within a process as a function of its distance from the soma, its longitudinal or geodesic distance away from the soma, we're not seeing any type of monotonic behavior. We are seeing heterogeneous and variegated calcium dynamics. So this is one feature that we want to consider when we start to look at the activity within these cells. Another feature is the propagation of events within these cells. So in the bottom row, looking at this figure in the bottom right, we can see again a soma with two processes in the astrocyte, and we can see events originating from process 1, and in the right-hand column, events originating from process 2. The difference being that the events originating from process 1 do not break into the soma. They are not propagating and affecting calcium activity within the soma, while events in process 2 not only affect the calcium within the soma, but also propagate further into the other primary process. So this speaks to some type of diffusive barrier limiting propagation, and we want to understand what these are and what's causing this type of limiting propagation or difference between the origination of these events and where these influencing factors are spaced throughout the cell. Another thing to consider, another feature of these calcium events is their stochasticity. If we try to categorize events within different regions of these cells, we don't just see, as a readout of a neuron, an action potential, a fixed type of shape and amplitude, et cetera. We see a distribution of these characteristics. As we can see in these 4 bar plots here, the amplitude as well as duration, or even in this bottom row, the shape of individual events is varying. And this is showing us that there is some inherent stochasticity, as well as even along with, I should say, the stochasticity that you're seeing from the stimuli, the random input that is going in potentially to these cells. So we want to categorize what is causing this inherent stochasticity within these astrocytes. And we're going to break that down now. As we start to consider a model for them, we're going to outline what are the different features we want to consider. And one of those features is the anatomy of the cell. So if we look at, again, an example of a soma with some processes emanating from it, the anatomy is it's branched and it's showing a lot of complexity, which could be a potential source of that variegated intracellular signaling we were seeing. Additionally, we've had a lot of talks today about the interaction between the ER and the cytosol through inositol triphosphate, IP3, and we want to consider the distribution of these endoplasmic reticulum, of these ER stores, and not just the distribution, but their morphology and their shape and interaction with the cytosol as well. Additionally, we need to incorporate the different calcium fluxes such as calcium sources, again, through the IP3 receptors, as well as agonist receptors linking the cell to external stimuli. And we also want to consider these sinks, these fluxes of calcium out of the cytosol through different pumps, as well as buffering, which could be a potential driver behind that propagation of calcium signals. There are multiple other mechanisms we could consider. There's mitochondria, which have an influence, and we saw that in an earlier talk, the influence of mitochondria in these cells, and as well as other mechanisms, but we're not going to focus on that. We're going to focus on the anatomy as well as some of these major sources and sinks affecting the dynamics of calcium within these cells. So the way we want to break these cells up and to consider this spatially varying morphology and spatially varying physiology is through this compartmentalization, whereby we can take an astrocyte branch, as you see here in the top right, and we can divide it into coaxial cylinders. And by that I mean each compartment is going to have an external cylinder representing the cytoplasmic space, as well as a concentric internal cylinder representing the calcium store, the endoplasmic reticulum. And in this way, we can vary across compartments the relationship between the ER and the cytoplasm. And in order to consider this relationship, we have to constrain it, and we want to use some influential work out of Alexei Semyonov's lab in 2013, which gave us a great way of characterizing this relationship between the compartments, or in this case, the surface to volume ratio of an astrocytic profile to the fraction of the calcium stores that exist in that area, such as in this case the ER. So we can use this to then fit a morphological relationship using an alpha function on this data, and this gives us a representation of the percentage of ER in a given compartment based off of that compartment's surface to volume ratio. To tie this all together, that morphological aspect, we also want to include this really important work, which Maurizio has had a lot of influence on over the past several years with his incorporation of IP3 dynamics. And we're going to also add in this, as has been done in previous work, this interaction between the cytosol and the ER through that morphological relationship on the previous slide. Additionally, we are going to be connecting these compartments through this type of diffusive flux between concentrations across compartments. This is a model that I have used a lot in the past, both for publication as well as for my doctoral work, and I want to break down a little bit the way we get to this diffusive term and how we can connect these compartments. And we're going to start with some compartmentalized astrocyte. We've taken our cell, we've taken our astrocyte, and we've divided the regions into these compartments, these cylindrical compartments, which need to be connected. And we want to consider two different types of connections, this consecutive connection as well as this branching junction in green here. So let's start with the simple case of 2 or 3 linearly connected cells, consecutively connected. We can take some inspiration from the diffusion equation, the classical diffusion equation, and we can look at a coarse-grained representation of that from the perspective of our compartmentalized astrocyte, where we can look at the flux of calcium across one of these membranes by considering the representation of concentration from the left and from the right. Once we have this discretized flux through this membrane, we can start to play around with the varying distances and the varying morphology. And one way we want to do this is by incorporating the area of the two intersecting membranes. And in order to incorporate this area, we are going to be changing units from this concentration per unit length to per unit area. And this allows us to get the area of this varying area across compartments into the equation. And with a little bit of algebraic magic, we can arrive at this ratio of areas between consecutive compartments. And this is allowing us to relate and incorporate the morphology across compartments. This formulation, which in this case is going from 3 compartments of CI-1 to CI+1, can be extended to a branching junction where we have one compartment branching off into 2 sibling compartments. And that's simply done by taking that ratio between the resulting branched compartments and iterating over the influences of those concentrations, as we can see here in this green branching junction, which we originally saw in that compartmentalized astrocyte earlier. So we've developed this compartmentalized representation of calcium dynamics within the cell, and we are incorporating this morphology within these compartments as well as connecting them through this diffusive flux. So now we can start to look at some of the results which we developed for the Fulbright application. And we're going to start simple with a simple structure of a soma and one process, where that process has 2 compartments. A proximal compartment and a peripheral compartment, which is also receiving some type of stochastic input. And we can break this into 3 tiers of our structure: the soma tier, that proximal tier with the 1 compartment, and that peripheral tier with, again, 1 compartment which is receiving input. And we're going to be characterizing calcium activity, calcium dynamics within these compartments by keeping count of the time, the spike times, or the times of calcium elevations, the saline times of calcium elevations, and then binning them, or binning the wait time between these spikes to be able to represent the activity within a given compartment. And here we see that without a lot of variance or complexity in our simple structure, the distribution of activity is relatively consistent across the three tiers, from the soma to the peripheral input receiving tier. But what happens if we start to add branching junctions, those branching junctions that we had discussed before? Well, we introduce complexity, and therefore we start to introduce some interesting dynamics. Specifically, I want to call your attention in this branching junction for the peripheral tier. We have the emergence across this branching junction of a bimodal peak in the ISI distribution. So by adding this branching junction, we have added another type of calcium event in this process, this wait times of around 6 seconds here. And additionally, if we move that branching junction closer, we see again a peak here with the proximal branching junction, a peak in the soma ISI, or I'm sorry, a bimodal peak in the soma ISI. So this is already starting to show some interesting dynamics just with these two simple structures, but we can take it a step further. We can add branching junctions to both tiers. This yields this relatively more complex than the other simpler structures, this structure of a soma with a branching junction and then another branching junction, which we'll denote as S2 and 2, S22. And we can see immediately here a change in the differences between the ISI distributions across these three tiers. Namely, this decrease in soma activity, from showing some kind of unimodal peak to almost uniform distribution of very long wait times as well as shorter wait times between calcium events. We see again this branching junction has caused this emergence of a bimodal distribution of ISI here, as well as these unimodal distributions in the tier 2 compartments, these peripheral compartments. And another thing to point out in these peripheral compartments is that C2, the input receiving compartment, the red compartment, its activity is marginally different than the other tier 2 compartments, which are not receiving input and are connected indirectly via other compartments. So this is just one example of how this has gotten more complex and starting to show some variegation. But another thing, and I think an important feature to point out for this structure specifically, is that we have introduced variegated signaling across our hierarchies, across our different tiers of astrocyte compartments. And what I mean by this is that we're seeing two types of differences as we move between tiers. We are seeing that a distance, and I should start by pointing out this table on the bottom left-hand side, which is allowing us to compare our input receiving compartment C2 to other compartments within our different structures, the simplest original structure, as well as those two slightly more complex structure, and then the current structure on this slide, the S22 structure. And we see that, firstly, as we move our Manhattan distance, or our connected distance, away from the input receiving compartment 2, we are increasing the difference between the ISI distributions. We're seeing more difference based off of how far away these compartments are from this input receiving compartment. And additionally, something else that's shaping the variegation of this activity is the morphological profile that we have introduced, which is changing from tier to tier. And we can see that the difference between tiers is also showing a higher degree of difference in activity versus in between differences of the same tier. So these two features are pointing towards this overall variegation of calcium elements or calcium events within these different compartments. And another topic that I really want to focus on is introducing this varying IP3 diffusion and how this affects the system. So in this slide on this figure, we are taking our S22 structure, and from left to right, we are increasing the IP3 diffusion. So starting with lower diffusion, we have a lot of separation of ISI distributions within the same tier. So a lower IP3 diffusion is causing more differences between these distal compartments, which have, you know, the same morphology, which have the same structure. While increasing the IP3 diffusion is leading to a more of a synchronization of the ISI distributions within the same tiers, the soma, the proximal, and the peripheral. Another impact is of the increased IP3 diffusion is the introduction of this bimodal distribution in the proximal tier. So it's introducing higher IP3 diffusion is introducing a more salient bimodal distribution, or the more defined types of activity within this proximal tier. And these two things point to the potential importance and the effect of IP3 diffusion on the propagation of signals, and as well as the variegation of signals within these different structures. So to conclude, I want everybody to leave with some takeaways, which is firstly that morphology and physiology is shaping the compartmentalization of these calcium signals that we are seeing. And this is something that we were showing that we saw in the experimental work, and we have been able to show a little bit of in this, even with these simple compartmental structures. Additionally, moving into that morphology, as a specific feature of that morphology shaping compartmentalization is the idea that branching junctions are accounting for different signals as we move either further away from the input receiving compartment to the soma, or closer along the astrocytic tree. And another thing that is happening that I want everybody to leave with is the idea that IP3 is controlling the separation between these different types of distributions and characterizing a degree of difference between calcium events within different subcellular regions of this compartmental model. Thank you.
Thank you, Evan. Very nice talk. Very cool results. So I will now proceed to start reading some of the questions, okay? So the first question is from Carlos Vivarios, who is one of your colleagues, although across the ocean. So Carlos is asking if you can say anything about what is the minimum diameter of the compartments that we use there in order to simulate calcium oscillations and calcium peaks consistently, and whether you could envisage more or less if you can use this model to address eventually the fact that the astrocyte has a spongiform morphology rather than just a tree morphology as you define. So what would you answer?
Great question. So firstly, I would have to say that I haven't investigated yet the kind of the borderline morphology of these cylinders as far as expressing different calcium events. Or at least I haven't expressed it. You can definitely see that as you start to decrease the size of these compartments, that you see the ability to express dynamics disappears. But we haven't, and it's something I think we're going to be looking into, is how the changes in morphology and the minimization of the radius, or the interaction between the ER and the cytosol, is going to be affecting calcium dynamics. That's something we want to look at down the road. And the second part of that question was, I believe, how to start to represent the more the peripheral parts of the cell as opposed to this tree-like structure. I would say that given this current model, it's beyond the scope. But as we start to incorporate more types of calcium dynamics, I think that'll become a very interesting question that we can look at. And I haven't thought much about how we can start to express this branching off into almost like a sponge-like activity. I haven't thought too much about that, but I'd be very interested to hear thoughts on how we could go in that direction.
Okay, thank you. You have another question by Rudiger, who was speaking earlier today. So the question is, if you change the total calcium flux per compartment, okay, as a function of compartment size, okay.
Right.
Wait a second, because I'm missing the question. Sorry. Do you change? Sorry, no. So okay, I was asking. So yes. So well, I can answer him, but you can also say. So Rudiger is asking essentially if we are scaling calcium flux by the volume of the compartment.
Yes, and I kind of threw those jumble of that diffusive flux, characterizing that diffusive flux. The idea is that we're incorporating the area, or in this case, the difference between area of consecutive compartments as far as scaling the flux or the influence that one compartment has on another compartment of a different size. So we are incorporating that in our diffusive flux term, which is connecting all of our compartments.
Okay. Any further question? Okay. So thank you, Evan, very much for the nice talk. And I will invite now on stage the last speaker of this fantastic workshop, Amit Agarwal from Berlin, the Heidelberg University in Berlin. And please, Amit, join in. Okay, Amit, let's try to say hello. How are you doing? Very good.
Good.
So let's try to see first if you're able to share your screen, and then I will proceed with the introduction. Amit has started more or less by now, I guess 15 years ago, perhaps, with doing his undergrad and grad training at Max Planck Institute for Molecular Medicine, Experimental Molecular Medicine, and the August University in Göttingen, under the supervision of a prominent molecular biologist that is Klaus-Armin Nafe. So then he moved to Johns Hopkins, and eventually in 2017 he came back to Germany and Europe, where he took over a position as principal investigator at Heidelberg University and at the Institute of Anatomy and Cell Biology. So he's the recipient of several prestigious awards, including, well, from early times, the Barry Hood Jr. Young Investigator Award, the Anuvardhana Rao Memorial Award, more recently a Brain and Behavior Research Foundation, also called NR-STAT, Young Investigator Award, and the Kika and Heinz Saller Research Group Leader Award. His work is mostly, I say mostly because you never know what is the future reserving, but is on neuroglia interactions, and in particular on calcium signaling. And we owe to him as well one of the first studies in vivo on the study of calcium signaling in mitochondria and their contribution in astrocytes. And with that in mind, I leave the stage to Amit, and looking forward to your talk.
Thank you, Maurizio. It was really great. So first, I just want to make sure that you can see me and you can also see my screen. So is that the case?
Absolutely, you can go on.
Good. Yeah. So because it is difficult to know what is going on. Great. So thank you for inviting me for this fantastic conference. I really, truly enjoyed it. So thanks, many thanks to all the organizers. Actually, Ivan's talk was a very nice primer for my talk, because I will be touching base to almost most of the things he talked about, but more experimental. I'm an experimentalist and less of a modeler. So, let's start with the astrocytes. So, astrocytes are very complex cells, which we don't have to reiterate. It was really beautifully shown in Leonid Svitsinko's talk yesterday for when he was modeling Astro, and also in Alexei Simenov's talk and several other talks, like also Ivan's talk, that these are highly complex cell, if not the most complex cell in the brain. And the complexity was really well understood and studied. But most of the study in astrocyte, in terms of calcium modeling, in terms of understanding the nature of the astrocyte physiology, has been done on cultured astrocyte, which look quite different. They are different kind of cell. They are definitely astrocyte. This is an image of a human astrocytes in culture. And if you look at this humble-looking mouse astrocyte, here is an astrocyte expressing the red fluorescent protein, TD tomato. And we can already see that they make these not only branches, but very fine spongiform-like processes. And this was already appreciated more than 30 years back by Hama and Kosaka using high-voltage electron microscopy. So in this image, you can see that the astrocyte is less of a soma and main processes, but it is full of these very fine branches. And these fine processes make contact with synapses. As you can see in blue, these very fine processes of astrocyte, they touch the pre and post-synapse, pre-synapse shown in green, and the post-synapse shown with this post-synaptic density in red. And these processes can be as thin as 40 nanometers. And that is a place where a lot of calcium exchange happens. And many times in these cultured astrocyte configuration, we will not be able to know at the business end of the cell what is going on in terms of how the cells are communicating. And the same thing goes also for the blood vessels. At the lumen, you can see again the process in blue. Here, marked in B, that this is an astrocyte process. They form the blood-brain barrier. And again, this is the business end of the cell where they interact with the neighboring cells, given it is a blood vessel, or even they interact with oligodendrocytes. So here what I'm trying to get at is that these cells communicate at the very fine processes and not much with the soma or the main process. So we tried that, can we understand or look at the calcium at these processes? So for example, here, if we take an example of a synapse, so here we have a pre and a post synapse. I think now I got my cursor somehow. So here is a pre and a post synapse. When it releases a neurotransmitter, the calcium goes high up in the cell. And this is again a very old video from Steve Smith's lab, Cornell video. Many of us have already seen it. First time to show that how a puff of glutamate on astrocyte, cultured astrocyte, leads to these calcium waves, and also beautiful calcium transients. And the bigger question here remains is that what are these, when do these calcium transients rise within the cell? And how are these calcium transients regulated? And to begin with, what are these calcium transients? What does this calcium transient mean to a cell or to a neighboring cell with which they are communicating? So these are much larger questions, which we will try to, it will take probably my whole career or many careers to able to understand that. But with that aim, we decided to look into the finer details of the calcium transients. So we use GCAMs, and we use transgenic mice, which express GCAM specifically in astrocyte. Here you can see that the GCAM is expressed in a single astrocyte. One little dot on the screen, what you see is a single astrocyte. In this image, which is a zoom-in image in the hippocampus, the labeling shows GFAP, which is the very standard marker for astrocyte. And these are membrane-anchored GCAM astrocyte puffs. And we can already see that with GFAP staining, which shows the major branches, we hardly see any part of the cell. Almost 8 or 9% of the cell is visible. This is a zoom-in image of one of the astrocyte confocal Z-stack to show that our sensor, which is tagged to the membrane, so we have membrane-anchored the GCAM3, which is a genetically encoded calcium sensor. So now these sensors can go to the fine astrocyte process. As you can see here, in the center, that circular structure is a soma, and that is not filled up with GCAM, as should be the case for the membrane-anchored protein. So how does the calcium transient look like in these astrocytes? So here on the left, you see astrocyte imaged in brain slice. Today, I will be presenting only brain slice data, no in vivo data. And here, when we do the imaging in the presence of TTX to block the neuronal activity, to isolate the astrocyte activity, we developed an algorithm to analyze these calcium transients, which happens in a very restricted pockets called microdomain, hotspots, any spots you can call, or people have given different names. And we can isolate these calcium transients very nicely using our algorithm, which we call Cascade. It is available, it's published, so whoever is interested should feel free to use it. And when we apply norepinephrine, for example, it's a very potent stimuli for the cell. You see that the whole cell comes up and immediately it goes down and goes back to its calcium oscillations, what it was going through. And this was very unique for actually norepinephrine. Other neurotransmitters, when we apply, like for ATP or DHPG, does not give this kind of effect. I'll come to it a bit later in my talk. So this particular cell, when we image for 5 minutes, this is, we pull out a lot of active regions. We call it microdomains. And here I've picked up like 10 microdomains, and we can see we get very beautiful signal to noise ratio. There is a whole diversity of the calcium transient. And here I would like to bring your attention to, for example, 2 domains which are close as 3 or 4, domain 3 and 4. They are very close to each other spatially, probably a couple micrometer, but they behave very differently. The calcium transients within these pockets are very different. Also, same thing for 8 and 9. They have very different set of calcium transient, telling that the anatomical location or how these domains are anatomically located will also control the diffusion and also the calcium transient, though they look very close, but they might be very restricted connectivity between them. So this plot of all these domains which we picked up, 125 of them, we see that there is not much of a pattern. We don't see a calcium wave within a cell going from one corner to the other. These transients tend to be very stochastic and also very sparse. So for example, if we image a cell for 5 minutes, and most of the domains here we have accumulated from 114 cells, more than 8,000 events, that 70% of the event in a given domain are just once in 5 minutes, which is very rare. Only few domains which are really, really low, as you can see, can have like tens of events. And this was also the case for soma. So this is the case for soma, that the somatic events are also very rare. And that always made people think that astrocyte does not participate actively in the neuronal processing. But this is one single plane of an astrocyte. And if we think about an astrocyte like a puff and do a back of envelope calculation, there might be 8 to 10,000 of such domains. And even the active ones, you can already calculate that a given astrocyte will be active all the time. And this was very beautifully shown by work of Andrea Volterra and Erika Benducci in the 3D imaging, that you can see that kind of activity ongoing in an astrocyte. Here, one thing which I wanted to bring is that many of these domains, they have this sparse activity, but they can also come together to have a much larger event, which you also see. So for example, here highlighted like event domains like 1, 2, 3, 4, 5, 6. They at one point, at one given time, they come up together, but afterward they are doing their own kind of stuff, showing that it is not that they are only segregated, but a larger calcium event can also induce calcium in the neighboring domains. One thing which was already appreciated a long time back, also discussed by Alexei Simenov yesterday in his talk, was that most of these calcium transients, the spontaneous ones, are not blockable by blockade of basal synaptic transmitters. So for example, a combination of baphromycin and TTX blocks the basal synaptic transmission, but it does not block these spontaneous calcium transients in the astrocyte, indicating that there might be other source than only the neuronal activity within these cells. So astrocytes have spontaneous calcium transient, and there are induced ones. So they can be induced, for example, by putting neuromodulators or neurotransmitters. Here, for example, norepinephrine. I showed you in the video, when we apply norepinephrine, there is a very locked, very simultaneously, most of these transients come together. There is a big diversity, as you can see in this correlation coefficient plot, that some cells have almost 80% of the cells, microdomains coming up together on application of norepinephrine, but roughly 50% of the microdomain comes up when we apply norepinephrine. But this is not the case when we apply other agonists. Very potent one like ATP, purinergic receptor agonist, does not do so. And also DHPG, which is an agonist of group 1 mGluRs, group 1, or mainly mGluR5s, which is expressed in these cells. The other thing which we tried to do was that we said, okay, great, when we apply repetitively within the same cell, different kind of neuromodulators, so activators of different receptors, for example, norepinephrine for adrenergic receptors or ATP for purinergic receptor, it generates different map, thereby indicating that probably the distribution of different receptors on the astrocyte should also be considered. It is not that everything is homogeneous. the somatic information tells us that, okay, great, when you apply many of these agonists, the soma always comes up, but this is not the case for the neuromodulators or other activators, which is released by neurons or other cells in that case. Here on the overlay, you can see roughly 50% of the norepinephrine and ATP activated microdomain overlap. When we do the same thing with ATP and DHPG, the overlap is far less. It's roughly 35%, indicating that we have to consider the heterogeneity of the expression of these receptors within the cell, which itself can lead to different modalities of calcium transient. IP3 receptor 2, we heard a lot about IP3 receptor. IP3 receptor 2s are the main receptors expressed by the astrocyte. There are literature people talking about 1 and 3 being expressed, but at very, very low level, but they are main receptors. Almost 12, 13 years back by the lab of Ken McCarthy, it was shown that when you knock out IP3 receptors, the spontaneous as well as the evoked activity is gone. With that idea, we did an experiment during that time that we blocked the Circa pump. We soaked the slices for 1 hour to empty the internal stores, and we saw a reasonable amount of calcium transient, spontaneous calcium transient within the astrocyte in slices. And that was a bit surprising given the published results. And when we started digging into the literature, we thought that maybe now we are seeing many, many more calcium transient in the final processes where probably hapsigargin does not reach to the highest concentration. So we decided to go and generate a triple transgenic mice, and namely IP3 receptor 2 combined with our double transgenic mice where we image calcium transient. So the knockout of calcium transient in these mice with knockout of IP3 receptor 2, we were kind of surprised to see that these cells, astrocyte here again, had significant amount of calcium activity. This activity is reduced, as you can see when you compare on the left-hand side the panel where you have spontaneous activity. This activity, the number of microdomains and also the frequency of event is reduced significantly by 2/3 or up to 70%. But one thing is there that when we apply norepinephrine or, matter of fact, ATP or other agonists, you don't see this activation. So the spontaneous activity persisted, a reduced level, and most of the evoked activity was gone in these cells. So now the question arose was, what is the source of the spontaneous activity within these cells and what we have been missing? So to cut the long story short, the indication came when we were doing this electron microscopy, so transmission electron microscopy using silver intensified immunogold labeling. So now, since we were expressing a membrane tagged protein, GCAM is a modified GFP, a membrane tagged protein, we thought, can we now look at the astrocyte processes, fine processes specifically of the astrocyte, and using the immunogold labeling, when we label GCAM, we see fine processes of astrocyte, as you can see marked in red. And one thing which became very apparent was these processes were filled with mitochondria. For example, in the zoom in here, you see that some of the processes have the mitochondria just almost stuffed into it. So much so that almost GCAM is stuck almost on the mitochondria surface, it seems like. As you can see here on the top image or also in the lower image, that these processes, the astrocyte process very close to the nerve terminals, as you can see, has mitochondria there. So it clicked that, oh, mitochondria can be a source as well as the sink for the calcium. So can it be that the remaining activity, which we are seeing in IP3 receptor 2 knockout mice, are generated by mitochondria? So we generated to design and develop a new mouse line where we can express mitochondrially tagged EGFP specifically in astrocytes. Here it is a triple transgenic mice expressing cytosolic TD tomato in the astrocyte and mitochondrial EGFP in the astrocytes too. And here you can see this is a single cortical astrocyte, this labeled with GFAP here, that this cell is filled with mitochondria. We did recently a quantification in a single cell. A single astrocyte can have, a cortical astrocyte can have up to 1,500 to 2,000 mitochondria. We are now studying the mitochondrial dynamics more in detail. I will be not talking about it, following in vivo imaging, and see that what does this mitochondria architecture mean to the cell and how it can be involved in regulating the calcium transients. So coming back to, do these calcium transients, which we see, co-localize with mitochondria? So when we do the measurement of the calcium transient, here mitochondria EGFP is pseudo-colored for ease in seeing. Most of these calcium transient do co-localize with mitochondria, up to 90%. So 90% of the spontaneous calcium transient, which we see, overlaps very beautifully with mitochondria, indicating that mitochondria might have some role to play. So we said that if we can go and close all the pores, whether it is MCU or whether it is membrane permeability transition pore or sodium calcium exchanger, can we block these remaining spontaneous calcium transients, which we see in IP3 receptor 2. So again, to cut the long story short, when we blocked the mitochondrial permeability transition pore by cyclosporine and rotenone in the slices, we see that virtually all the remaining, or most of the remaining IP3 receptor 2 calcium transient in IP3 receptor 2 knockout mice were gone, indicating that they were being generated by the transient flickering of membrane permeability transition pores on mitochondria. So now the question arose is well, this is the exit path, as you can see, MPTP. Then there is an entry path to the calcium also. And the main entry part for the mitochondrial calcium is MCU, mitochondrial unipolar. So that time we raised a question that can we see now the spontaneous calcium flickering, which we see in the cytosol, also within the mitochondria? And if we evoke a large calcium rise within the cell by, for example, applying norepinephrine, can we see this calcium channeling in the mitochondrial matrix? So for that, we designed a AAB, and we drove it by GFAP, minimal GFAP promoter, and tagged GCAM6S with mitochondria tagging sequence. Again, imaged in the acute brain slice from the adult animals. And here we see that there are spontaneous flickers in the mitochondria. And when we apply norepinephrine, there is also like a synchronous activity within mitochondria also. As you'll see that a lot of mitochondria will all come up together, indicating that not only the cytosolic calcium transient we see, but this also dictates into mitochondrial matrix calcium transient. So to analyze the entire set of data which we are acquiring, we have developed, revamped our old cytosolic calcium transient analysis method for analyzing the mitochondrial calcium signal. They turn out to be of very different characteristics, so we had to revamp it. I will not go into the detail. It uses the same kind of machine learning algorithm to really pull the signal and to classify the signals. So how do the signal look like? So if we see the spontaneous calcium transient, there are a big diversity of signals. Some signals are very large. They can last for a minute, as you can see in the upper two traces. But some signals are very fast, within a few seconds, almost like a cytosolic second, as you can see in the yellow or orange traces. And some signals can be basically recurrent, as you see in trace number 5. When we apply norepinephrine, there are many regions which were not active during the spontaneous activity comes up, as you can see from those red boxes. And the signal prolongs. Not only the intensity, as you can see in this heat map, the amplitude of the signal increases, but also the duration of the signals increases. For example, some of them can last for several minutes. As you can see from the scale bar there, that one of the upper blue signal is going beyond 3 minutes. So these cells can really take up, these mitochondria can really take up calcium and keep for more prolonged time, which is very distinct. The similar experimental setting, the cytosolic calcium goes down completely. Probably it is buffered very nicely by mitochondria and then slowly released by the mitochondria. So now the question is, this is still a hypothesis. We have not really seen it. So the idea is that can we simultaneously measure cytosolic calcium transient and the mitochondrial transient? So now we have decided that can we take use of the red mitochondrial targeted sensor. We already have membrane anchored GCAM for cytosolic calcium transient. So can we use or put the sensor within the mitochondria matrix? So we decided to go for JR-GECO1A. This calcium sensor is in the red, is derived from the red fluorescent protein, and has a characteristic very similar to GCAM6S, so it's very sensitive, and it has a low KD, as I said, for same like GCAM6S, and also it has 2 pKa values, which is very ideal for mitochondria, for example, because the compartment within the mitochondria can go very quickly from acidic to alkaline. So there's a lot of fluctuation, which is not very good for GCAM, like GFP-derived sensor, but is good for the red fluorescent protein-like derived calcium sensor. So first thing what we did was that we tagged this sensor to the mitochondria, these are in hex cells, to show that well, can we really send it to the mitochondria, or sometimes these sensors are finicky, so this sensor does go to the mitochondria. When we apply inomycin to increase the cytosolic calcium transient, we see beautiful calcium increase. So we have now designed the mouse lines. We have the mouse line, and we are crossing these animals to simultaneously image calcium transient in the cytosol as well as in the mitochondria, both in the living animals now very soon, as well as in the slices, to be able to understand that what are the dynamics and the kinetics of these calcium transients and how this interplay between the mitochondrial and cytosolic calcium transient occurs. So to summarize what I've been telling, so what we have been thinking about, and of course it is central to the mitochondrial calcium handling, the endoplasmic reticulum, for example, circa pump IP3 receptor 2, and also they are induced by a very complex plethora of G protein coupled receptor expressed by these cells. But we think that in the new model, we need to integrate mitochondria because it seems that the way they are located anatomically and how they handle the calcium immediately within the cells, they can really change not only how the calcium is handled within the cell, but also the characteristic of the calcium signals, prolonging the calcium signal. So for example, it might be that endoplasmic mediated calcium signals are much more faster, and it is just at the level which we study them is prolonged just because of mitochondrial intervention. So this is our model of looking at the microdomains of astrocyte, including both endoplasmic reticulum and mitochondria, and probably we will have much more detailed calcium data between how these two organelles talk to each other in the smaller compartment of astrocyte. With that, I would like to thank my lab, relatively new, we are 2-year-old. And most of these experiments of astrocyte is really put forward by a postdoc from Chile, Felipe Bodaleo Torres, and a very talented master student who has been developing the code. And we have a lot of collaborators where we are doing the structural analysis of mitochondria in collaboration with the Department of Stefan Hell and also in the Department of Klaus Nabe in Göttingen. And several of these lines and tools were developed while I was still in the lab of Dwight Burgos at Johns Hopkins, so many thanks to him. And also we are doing some pathological study with Agabo Pepsol at DZNE. And with that, I will be happy to take questions. Thank you.
Thank you, Amit. Very beautiful work and results. So we have several questions, I guess, as they are populated. Can you hear me?
Yes, I can hear you well, and I'm trying to do some. So should I just keep my talk in there? It will be better.
No, well, I mean.
Oh, stop sharing.
Yeah, it's okay. It's okay. Just keep the slide like this.
Okay, perfect. Okay. Yeah.
So in case you need to scroll down and up, it's gonna be okay.
Yes.
So the first question comes from Kirsten Lent, who you may know is from Tampere University, right, Kirsten? So she's asking whether the images that you are showing are only, or the analysis that you perform are only based on single plane images, or you had other images? And in particular, what would you say are the different sources of spontaneity when you show the images of stimulation by norepinephrine or ATP or spontaneous calcium? Could this be coming from different sources in the cell, or could be an artifact maybe of different imaging planes?
Okay, so there are several questions, I think.
Yeah, it's a convoluted question.
Exactly. So we start with Rai. Hey, Kirsten, thanks for the question. So I will divide into 3 parts this question. So the first part is that most of the imaging, is it coming from a single multiphoton plane, or it is coming from 3D images? So most of the images which I showed you do come from a kind of single plane, I can say. We image 4 planes for mitochondria, especially for the calcium transient, cytosolic calcium transient. We do only single plane imaging. So essentially, we are just sampling 1 micrometer Z plane of a cell, because that is a 2-photon, essentially rule of thumb, 2-photon cross section. So we are collecting data from only 1 micrometer of a Z-plane for cytosolic. In terms of mitochondria, they do move. So we collect the data from roughly 4 to 5 micrometer, and we collapse them together to make sure that certain mitochondria are not just running in and out, and that generates a pseudo signal. So that is kind of 3D. We are trying to image mitochondrial calcium transients are slow. We are trying to image the mitochondrial transient in 3D, but there are a lot of problems. And one of the problems is a registration problem. If we image the cell, mitochondria, or any kind of transients for cell for half an hour or so, we generate massive amount of data, and then to register them in 4D plane, X, Y, Z, and T, is not that trivial. So we are now giving up partly for our 3D project, but so short answer is most of the data is in the single plane, but we are trying multiple planes. Second was that other sources of calcium transients, the spontaneous one, quite likely, that you are interested in. So yes, there are other planes. There are beautiful work from Andrea Volterra's lab, as I said, also from Alexis Semenov's lab, recent paper from Christian Henneberger, Baljit Karp. So there are a lot of excellent groups who are looking into this question. And it seems that also the extracellular calcium does play a role. So when some of these labs depleted the extracellular calcium, they show that these calcium transients also vanish in IP3 receptor 2 knockout. But this can also be like also you're vacating the mitochondrial calcium. So it is a very complicated problem for which we don't have a very easy answer, but there can be surface receptors which might be involved in this. At least we did not find a major contributor, single major contributing receptor. So your third question is related to the activation, right?
Yes, exactly.
So that when you activate with some of these norepinephrine or so there, one thing which I did not understand was, what do you mean by artifact? So, well...
So the specific question is, I can read it to you textually, so as verbatim. So when you show the overlap of stimulation by norepinephrine and ATP, could it actually stem from different parts of the astrocytes or different imaging planes?
No. So this is, we can be sure that we are in the same imaging plane, because one thing which you did not see is that several times we also have a structural marker, TD tomato. And also we know when we image the cell that we have not moved too much on the plane. So yes, most of the data comes from single plane. These are, if not 100, but then probably several dozen of cells imaged. And also what we do is when we apply drugs, we apply the drugs randomly. We don't apply them in a particular sequence, just because of the same concern which we had is that of course, when you activate the cell with, let's say, norepinephrine, and then you activate with ATP, then maybe there is some kind of weird kind of receptor desensitizations, who knows? So to make sure that all these drugs were applied in a different order, first maybe DHPG, second norepinephrine, ATP, and then you can mix match. So from the toss-off point to make sure that this does not come from that. So we tried our best at our level to make sure that most of these artifacts, either plane change or some pharmacology, does not mark our result.
Right. I might have a question on that, but first I want to read the questions from the audience. So Alexei Brasse is following up, and he has several questions. So first of all, how stable or reproducible are the microdomain spatial maps subjected to multiple administration of the same drug?
They are very, very stable. Actually, this was one of the reviewers' question which we got, and we went and did.
I might be the reviewer. You never know.
Exactly. There you go. So this was one of the reviewers' questions where we did a couple experiments to make sure that was the case. So there are certain things to it. So when we follow the spontaneous activity for a long time, then it does seem that these pockets of activity has some anatomical basis to it, and hence they are reproducible. So they come up time and time again. When we apply, let's say, same agonist over several times, let's say first time we apply, let's say, ATP, we wait for another 10 minutes, wash off, we apply again ATP, we wash off for another 10 minutes, we apply again ATP. There is some receptor desensitization, but most of the similar domains can be reproducibly evoked. A calcium can be evoked in the similar domain. So it seems very reproducible. And now with our long-term calcium imaging, which we are doing in the animal for up to a month, looking at these domains in vivo in the single cell level, and there also seems to be a high level of, let's say, restriction that these domains seems to be very, not transient, but quite permanent. They change a bit, but they're there, restricted, telling that it is an anatomical basis.
And always Alexei is asking if you see any correlation between the microdomain shape and actual astrocytic morphology.
Oh my God, that's a very hard one.
Yeah, I know.
For that, there was a very nice paper recently published in Nature Communication from the collaborative work with several labs were part of it, but it was from the lab of Valentin Nagel. So they combined STED to see the astrocyte morphology. Astrocyte morphology, as you know, Alexei, is not very easy to look at even at the normal light microscopic level. So they combined STED microscopy with 2-photon imaging to image calcium dynamics. And it seems that they came up with that these tripartite synapse, that there are certain nodes where the calcium transients are more often than the other places. So yes, people are trying it. We haven't tried it yet to combine both of them.
Fantastic. Very, very nice work. So I guess if the audience is coming up with other questions. Okay. So no more questions. Amit, thank you very much again for your talk and your contribution. And I am going now to shut down. No, well, Ivo is going to kick you out. You're not longer required. We are saluting and thank you again, all the audience, our speakers for these 2 days of. I hope you enjoyed these. I think we did as organizers, although we were between through following you and doing a lot of technical inquiries and texting behind. I think it was fantastic from our point of view workshop, and we are very thrilled to have coordinated this effort, bringing together people and investigators from seemingly disparate fields that, however, you see, when brought together, they can cross-contaminate ideas on such transversal topics as calcium signaling that is really ubiquitous. And I would like to thank the organizers and co-organizers, Ivo Sitman, that is on the other side of the half of the screen. Thank you, Ivo, very much for your support and help. Teng-Shin Kao, who is by now, unfortunately, the time zone difference is not in favor of him to be with us here. And along with that, also the main meeting organizers to allowing us to showcase this workshop, and as well as the technical volunteers and supporters and the technical staff that was able to always back up on us and save us in some of the difficult situations we had over this past 2 days. And I guess also for those of you that are not aware, they did a huge job in order to set up this main meeting and these workshops, facing sometimes harsh criticism and outbursts of frustration with technological platforms directly from some of the organizers. I speak for direct experience, but they excuse me right now. And again, thank you very much for your participation, and then to the next time. And thank you. Bye.
Thank you very much. Bye-bye.